Hi, so good morning. My name is Egon Trujillo, and today I'm going to be presenting to you my UDL lesson plan that I have implemented in my class this semester. And so I just wanted to give you a brief overview of what my plan was, how I designed this lesson, how I implemented it, and finally how we're still working on it and almost done completing it. So I'm going to first just give you a little brief introduction to the lesson. So the lesson I've been working on with my ninth grade biology class has been to start to begin understanding the structure and function of prokaryotic and eukaryotic cells. And so the specific lesson goals and more the outcomes that I had and objectives for how to accomplish this has been kind of spread out over a period of almost 2 weeks now. We've been doing this. So one of the first activities that I like to have students do when I'm presenting cell structure and function, just because there's so many of them, is give them different kind of examples of them rather than me just showing them notes on cells. And so one of the first objectives that I had up was students will be able to individually watch brain pop videos titled Cells, and I'll show you that video in a sec. And then I use Cornell Notes strategy and 5E lesson planning in my design. So what I have them do after we take notes on videos is I have them just kind of record some observations, ask questions about what it is that they saw, and then just give me a summary of what they learned. And I am very flexible with that, and I'll show you some examples of how I allow them to kind of just tell me or represent to me what it is that they think they got out of the video. And we use that Cornell Notes strategy to accomplish that. And so I'll be showing that in a bit. Then from there, I move on to having students like the next day or so do like a think, pair, share, just because the previous day we had watched that video and kind of just getting them to think about what cells are, what defines cells as living things. And so what I have them do with this think, pair, share with the buddy that they sit with is just to kind of begin discussing with them and showing them specifically how they think that cells are alive through their ideas. And I'll show you that in their notebooks in a sec. Using what we had discussed previously and the unit leading up to this, which is on cell theory. Then from there, the next day, I had them work with a partner again. And I like to use, rather than text, I like to use a lot of digital tools in the classroom in particular. For my two focus students, I like to use the Cells Alive and Brain Pop in particular, just because these two websites offer videos, they offer animations, tutorials, and they kind of allow them to just discover more about cells rather than from just a simple text. And because they are interactive, the Cells Alive in particular allows them to label corresponding cell organelles belonging to each of the major cell groups we've been discussing, which are eukaryotic and prokaryotic cells. And from there, they use those websites like Cells Alive to begin to start labeling the cell organelles, the major cell organelles we've been talking about in this class. From there, what I have them do, and this is about day 3 or 4, I believe it was, I had them individually now draw, create, represent, however it is they want to express their interpretation of what cells are. And so they make me a model of those cell types, and I have them use some of my 3D models of cells that I'll be showing you in a bit. I do have some students that just, don't like to get up and interact with models, so I do provide textbooks for those that just prefer sitting at their desk and reading. I do like them to use Cells Alive and any other animation tutorials that I do suggest students use for those students that prefer the animations, just because it provides labels, it actually discusses function for them using the text-to-speech feature. And so I think those are really good options to give students who aren't as comfortable with the language. In particular, my two focus students, these websites prove very useful for them so that they could begin to illustrate cell organelles and discuss their functions on those worksheets that I'll be showing you, and the functions of them as I had them then organized onto separate charts, which I'll be showing you. And then going on to the later part of the lesson, kind of getting close to where we are now, I had them then individually use that knowledge on what they learned about cell structure and function to compare and contrast cell types, cell organelles, to what they then put on these eukaryotic and prokaryotic cell organelle charts. And then this is where we're at now, kind of tail end of it now. We're using microscopes, scientific tool for observation that I like to expose them throughout the year with. And they're using these microscopes to analyze and then identify those cells on unknown cells. So they now use previous knowledge. Everything has been leading up to this. So now that they've been able to create their models of their cells, label structures and functions of cells, and whatever medium works for them, be it drawing, be it reading from the text, be it looking at my 3D models, they can then start to defend their answers in terms of what cell they think they are looking at. Is it a plant cell? Is it an animal cell? And they're using what they know about cell types and cell structure and function to do that. And so from then, I am moving on to the outcomes for this lesson. So what I specifically am embedding for this lesson is one of the NGSS standards, which is about structure and function in particular, and it's developing using models to illustrate hierarchical organization of interacting systems that provide specific functions. So we've been looking at the structure and function again of cell organelles, and not just multicellular organisms, but also in bacteria. So after having done my two interviews with my two focus students, I was able to then figure out what are the specific student needs that they will require to kind of break down that learning barrier for them. But I also want to make sure that all students' needs are addressed here. And so I like to give options for science, just because science is more of a class where the learning of it is kind of just giving students options to think in science. They need to be able to talk science. They need to be able to do science all the time. And so I believe now by using UDL as that tool, I have created for not just my focus students, but all my students a flexible curriculum that's inclusive through giving students these options that I'll be sharing with you. And these options now are giving students ways to interpret science. They can now participate through discussions about science. And of course, they're able to use science through the activities, labs, and hands-on stuff that we do, not just in this lesson, but I believe now for future lessons, this will be really helpful in my class. So ways that I think they can think in science. So by exposing students to the scientific process through practice, reading, discussions that we have, practicums like this identification lab, they can then start to learn how to think like a scientist, not just common sense, but more evidence-based argumentation is what I'm having them do. And so for my first focus student, Jefferson, I found that giving him hands-on options and extra help as needed, which he specified to me in my second interview with him. He says he's a kinesthetic learner, so he prefers doing hands-on activities. For my second focus student, Betsy, she shared with me that she loves to talk, she loves to act, and so I believe that giving her just as many options or opportunities, I should say, to let her discuss, let her talk, let her share, and even act. She says she loves acting, and so giving her a chance to act out what she's doing is going to be really helpful for her. And these are options I would provide them and any student who prefers it. talking in science. So in order for students to be able to actually practice science and do it, they need to be able to talk it. So they need to be exposed to scientific vocabulary daily in preferred mediums. And what I mean by that is, be it through discussions, be it through posting stuff through our discussion forums, which I'll show you, be it through just drawing and illustrating for me in their notebooks, scientific vocab. And so for my first focus student, Jefferson, again, since he likes hands-on activities and has requested extra help, giving him the opportunity to use his iPads, which has the text-to-speech feature, and also the textbook is available on their iPad so they can read, or have it read to them, I should say, just to help support with his English, because he did share with me in his interview that his English, he's not comfortable with it, and he really, really wants some extra help with that. Betsy, of course, she told me that she is really proud of herself because she feels like her English has improved vastly since she first started with us at this school. And so she has asked that I give her as many opportunities to talk in English. And so this is going to be a perfect vessel for her. And lastly, they need to be able to do science. All students need to be able to do science. And what that means for me, I believe, is just giving them practice doing science through activities, through labs like the ones we're currently doing, gathering evidence, observations like we're doing. And for Jefferson, again, this is going to be a lot of just hands-on practice for him. And for Betsy, it's not just going to be doing science, but really just discussing, thinking out loud, sharing out with me, what is she actually doing? And so now I want to go over my specific lesson design, and for that I'm going to kind of walk you through some of the materials that I am having students use. So I'm going to go ahead and turn on my lights. I realize they turned off, so give me a sec here, sorry. Okay, so I do want to walk you through all of the multiple means of representation, multiple means of action and expression, and multiple means of engagement that I came up with for these two focus students and for the class in particular. So I'm just going to first start with what I did for this lesson, and then I'm going to walk you through actual examples for all three of the options I gave my students. So just starting with the lesson that we're doing on cell structure and function. And so according to the UDL framework and the CAS website that I've been researching, there are 9 specific guidelines that they suggest that we as teachers use as a toolkit. Not necessarily every single guideline is going to be featured in every single lesson, but it is a start. It is a way to kind of make the curriculum inclusive and flexible for all students. So what I did is I introduced the lesson to them, and I gave them some options for each of the guidelines, and I used the CAST website and the checkpoints kind of as my baseline to ensure that I was not only including my 2 focus students in the lesson, but giving opportunities for students, not just in the margins, but on the whole spectrum, a chance to really think, talk, and do science. And so the first guideline, which is giving students options for perception, what I did was I offered students a way to kind of customize the information that I display in the classroom. One of the ways that I did that, and I'm going to go ahead and start walking around now, is I want to show you that how I did that was I give students, well, the school, I should say, gives all of our students iPads here, and they all have a text-to-speech feature. And so when I am displaying notes like I'm doing right now, I ensure that all students have, if needed, that text-to-speech feature available for them on my notes for the PowerPoint. So I'll just take us back over here. I'm going to walk you through our classroom here. And so this is a student laptop. And I wanted to show you, if I log in here, how I do that with them. Well, while that loads, what I'll show you is, well, I just wanted to talk about, so for students like Jefferson, who has requested that he be given a lot of hands-on exposure, one of the ways that I can give him an option for perception is I give him some online tools, animations, and models to use in the classroom. So I just wanted to walk you over here and show you where we have some of our online resources available. So this is the actual student portal that I created for my students, and I'm currently logged in as one of my focus students. I'll just change her name there so it's confidential, but this is her actual student portal, and I create folders for all of them so they know where to find stuff. So handouts, assignments, and notes. So going back to how I give options for the way I display information. And so I give her a folder, well, I'll just blur the name there. So here are her notes folders, and these are the notes we were using this week for cells. And I have it downloaded for us to view here. So if you click on this, or if the student clicks on this, wrong one, sorry. If she clicks on the notes for cells, she has her notes available here. And again, for Jefferson as well, this is really useful because like I said, it has all of our laptops have text-to-speech feature built in. I have a lot of visuals here to display for him, for Betsy, and for any student. So what I like to do is just give them this alternate way of presenting information rather than just the textbook. So here's just a little bit on cell history and some of the things we've been talking about. Now, the next checkpoint for offering students options in perception is giving them alternatives for audio. So what I like to do is, rather than just me sitting up here lecturing for them, and I do have them watch videos, and so I like to use BrainPop a lot. I find it very useful. So this is the exact video that I showed them on cells, and it's really neat because it gives them a tutorial on what cells are, and all of the videos come with built-in quizzes, little activities that I can do with them. But really, it's helpful because it's offered in English and in Spanish. So for giving students the options for visuals, they can see animations here. I also like to use this website known as Cells Alive, and this one's really neat because this one has 3D models of each cell type. So if I go here, we've been studying, like I said, bacteria, animal, and plants. I'll let you see that. And when they click on it, it gives them not only all the function of every single cell organelle, but if they click on it, so let's say animal cell, for example, it actually is interactive, so they can highlight on a particular organelle. It highlights it, they can click on it, so there's a nucleus, and it gives them the function that it can also read to them. There you can see the little mic there. And so, again, to give more students options for perception, this is just one of the ways, or one of the options I offer. The next checkpoint is offering students alternatives for visuals, and again, this would be a great option for that because they can see what cells look like. I also like to give students the options of interacting with cell models, so I have a couple of cell models here to show you. And again, other options being the Cells Alive website, the BrainPop video. And then, so let's just go back to our next guideline for means of representation, and that is options for language and symbols. So this one really involves kind of like the language of science. And so for Guideline 2, Checkpoint 2.1, clarifying vocabulary and symbols, the way that I do that is in my notes, which I'll show you again. What I like to do is I highlight a lot of vocabulary that is essential for the lesson that we're on. I like to explain it in detail, but the way I have them kind of let them explain it for themselves is I do put up a word wall, which I'm going to show you in a bit. I always ask that they use the glossary in the back of the book, and then I also have an online glossary, and I use these biology flashcards. So let me walk over here so you can see that. So here is our word wall that we're kind of wrapping up. We're getting through cell organelles, and we're focusing on the membrane and the processes of photosynthesis and cellular respiration and whatnot. And so I like to give them these glossary, I should say flashcard terms, because they are interactive for students like Jefferson, who says he likes to just kind of pick things up and learn from it. So I like to give them those options. I like to, again, show them in the notes that all of my notes are highlighted with special attention to words that we're covering. So for example, we've been talking about cells, and I like to show them pictures, and I highlight things different colors so they can see what's really important. The next checkpoint for giving options for language is I like to clarify the structure of cell organelles, and so the way I do that is I like to, of course, give them these videos, these illustrated websites, and just have them kind of understand the structure of it rather than just reading it from a text, but actually holding it with our models, actually seeing it on these interactive websites. The next checkpoint, checkpoint 2.3, supporting decoding of text, what I like to do is I like to do what's called like echo reading. And so for students like Betsy, this is a great opportunity because she has told me that she likes to talk in English in particular because it's really helping her with her pronunciation and her reading. So I do a lot of reading aloud to support her decoding. I like to, of course, give them the option, if needed, for all students just to use that text-to-speech feature if they don't know how to pronounce, like this word here, amoeba. It reads for them. Again, I do put up a word wall, and I ask that they either use our flashcards or kind of create their own with definitions on the back to kind of just help support how it is they're learning these terms. Moving on to the next checkpoint, checkpoint 2.4, promoting understanding across languages. Like I said, one really neat feature about Cells Alive in particular is that it does offer, and you can see that, a Brain Pop Espanol, because my two focus students are newcomers to the country, and so they sometimes, Jefferson in particular, asks if he can have versions of things in Spanish. So that's one of the options I give for him when I am trying to promote understanding across languages. The other checkpoint, 2.5, illustrating through multimedia. Like I said, I don't just give, I try not to use the text very often. I like to give them my interpreted notes of the text. I let them use these videos and tutorials as options on their iPads or laptops if they want to learn. I also ask them to do drawings of cells with illustrations, which I'll be showing you in a bit. And of course, I'm using these cell models to help them learn more about cells and help them to illustrate their cell models. The last guideline, number 3, for comprehensions, I like to give them lots of options to activate background knowledge, and so that's where I have these notes available for them. We've been discussing cell theory previously, so we started talking about cell theory, and now what we're doing, like I said, is we're going to have them kind of apply their knowledge about cell theory and see if they can identify what cells are and organelles that make them either a bacteria, animal, or plant cell, and what makes them living. So what I did here is, of course, I gave them these notes on cell structure and function, which are kind of towards the end here. Then I taught them how to use microscopes as a tool, and I'll move over here to the microscopes now. I let them use microscopes as a tool to learn about cells. I, at the beginning of the year, taught them just kind of how to argue in science, how to do the scientific process. And what I do to kind of support their comprehension is I do a lot of warm-ups, and a lot of the warm-ups entail them having discussions about what it is we're doing in regards to cells, what are the organelles, and so for students like Betsy in particular, who likes to talk a lot, and so she learns best by talking, this is a really good exercise for her. The next thing that I do to support with the option for comprehension is I like to highlight patterns, features, and big ideas. And the way I do that is, like I showed you, I do highlight some vocabulary notes. I do put words up on the word wall, and I give them these nice sheets to kind of help them organize their thoughts. So when we're looking at cell structure and function, what I have them do is on here, they start to illustrate the parts of the cell. And this really addresses the next checkpoint, which is information processing. And what I have them do is they're able to kind of summarize all of the organelles on here. I let them kind of organize all of the structure and function of those organelles on these charts that I give them. And this is really helpful also sometimes when I let them do, we did a little pop quiz last week on cell structure and function, and what I let them do is I gave them the option for my period 5 in particular, because it's a sheltered class, I give them the option of using these notes, using these worksheets during these assessments, and that really helps with the information processing I found. And then lastly, to ensure that I'm giving options for comprehension, is I am making sure that I always maximize transfer and generalization of this information. So what students are doing is they're using the previous knowledge they attained from doing these worksheets, from doing these charts, and they're using it to actually identify cells under the microscope. I'm going to move on now to the next multiple means of. Now we're going to go to multiple means of, hold on here, sorry, my laptop froze up on me. Well, that's okay, I'll wing it. The next multiple means of action and expression, there are a couple of guidelines that I also use for that. And so the first guideline is for physical action, to ensure that I give all students options for physical action. And what I did here is I kind of varied the methods of response and navigation. And the way I did that is to navigate learning about cells. Like I said, I gave them this option of using these websites like Cells Alive that have these quizzes, so they can kind of assess themselves. They can actually respond using Cells Alive for like formative assessments, like little daily check-ins that I like to do with them. What I also like to do is they can interact with these models rather than just seeing it online or reading it in a book. They can actually pick up the models. And again, for my focus student Jefferson, that really helps him because he is a hands-on learner. The other checkpoint for physical action is optimizing access to tools and assistive technology. And I really, ask that students bring iPads every day, just because, they have the option of recording, photographing all these observations of cells. Some of them like to draw them, some of them just take pictures of what they're recording under the microscope. And I also offer a digital microscope that I project in class while I'm doing this, so that's really helpful for them. And then, of course, they have these interactive cell sites like Cells Alive that is, an assistive tool for them. Moving on to the next guideline, Guideline 5, Expression and Communication. I like to give options for that, and the way I do that is I use multimedia to communicate. So like I said, I do offer all my students notes in their notes folder, so they can always pull these notes up for reteaching or just for review. The tutorials and videos online are really helpful for me to communicate with them what we're learning. I then like to use a lot of tools to construct and compose what I'm doing, but also for them. So what I found really helpful is I like to let them, do their own drawings of what it is we're looking at to kind of show similarities and differences of things. And like I said, I do like to use these worksheets as kind of the baseline and these charts to let them see the similarities and differences between cells. And I find that just, giving them this as an example helps for them to create their own. So let me show you actually some student work I have over here. Sorry, walking around my lab here. So this is an actual student notebook. And what I like to do, like I said, is in order to make sure that our students are using multiple tools, I do these warm-ups. And so for this one, we were discussing a couple weeks ago, just kind of like what they think cells are and why they're alive and whatnot. And so this student right here, she went ahead and defended her answer with not just an explanation, but she actually drew some cell organelles to validate that. So again, giving them lots of options to represent their answers there and compose their answers. And I really find this is useful for building fluency with graduated levels of support. So I'm starting them off with the worksheets kind of as their baseline, and then that they move on to kind of defending their own answers with their own drawings. So and again, those websites really help to support with that. For Betsy, this is really cool for her because it helps her language acquisition to be able to express herself in any means she wants. So she doesn't have to, for example, draw or write me an answer like this student did. She can actually share out during these warm-ups, because during the warm-ups I actually give them options to think, pair, share with a partner. And again, they can draw, they can discuss what it is that they want to help with the building of fluency. Moving on to the next guideline, Guideline 6 for multiple means of expression. I like to give options for executive function, and one of the ways I do that is I actually like to have students kind of set their own goals. And the way I do that, and I'm going to show you here my actual standards board. So I do post, here are my next generation standards. So there is the exact standard NGSS standard we're focusing on, which is HS LS12. I also post the guiding questions for them. I like to post the science engineering practices for this lesson, and of course our cross-cutting concepts for this, all of which I'll provide a copy of when I submit this video to you. And a really cool thing that I do for these students, and let me walk you over here, I forgot to bring it, is I like to check in with them. And so in order to give them appropriate goal setting, is I like to give them what I call my goals for success, and this is something I have them do every grading period. So this one looks like it's from last month. What I do is I have all the students collect their signatures and grades from their current teachers, but I have them list kind of like why they believe they are attaining that grade, and then I have them kind of set a goal for themselves. So just tell me what it is that they think they can do better next time, and most importantly, what the parent can help them with. So this really helps to help them set their own goals. The next checkpoint in executive functioning is supporting the planning and strategy development, and what I found really helpful is, again, giving them these types of worksheets and interactive models online to kind of show them what it is each cell looks like, and that kind of helps them in their own mind kind of understand better, so that they can more strategically develop a clearer idea, so that when they're asked to defend in the final assessment, which is identifying unknown cells under the microscope, they'll be able to do that better. And of course I use the previous knowledge about cells and cell theory to help them know what to look for. The next way that I have them ensure they're performing executive functions is I facilitate managing of information. And what I like to do, like I showed you, is during these warm-ups, such as this one here, I like to give a lot of stop and think time, a lot of wait time on these warm-ups to really think about their answers. And so like if this student here, she didn't know what it is that makes a cell. Well, she can go back into her notes. So she has some great notes here on what cells are, what cell theory is, and that helps her with, kind of locating, and I have them highlight, actually you can see there, I have them highlight where things are, and I'm going to show you her table of contents. So, so here we go, notes on cells. So everything is color coded, so it helps them to identify where things are a lot easier. So that really helps with this whole, sorry, I lost my page here, helps her with identifying resources, and I also, like I said, like to make sure that they know where everything is in their notebooks. The next checkpoint for enhancing capacity for progress monitoring is I like to do things like this. I like to do my little warm-ups, such as this one, where I ask them a question, and in this one was a true-false about cells. So that really, for me, is a form of a nice little, it's a formative assessment where I just give them a daily stamp, and that way they're receiving progress, so they know how it is they're doing with these kind of daily check-ins. Moving on to the last multiple means of engagement. This is probably the most difficult in science, because if there's no buy-in from the student, then it makes it very difficult for the student to be able to actually do science, talk science and think in science. So going back to my focus students, Betsy and Jefferson, like I said, what I tried to do through my interview with them was kind of get to know them, who they are personally, academically, and kind of how I can support them based on what it is that I know. So in order to address Guideline 7, which is recruiting interest, giving options for these students, I really like to give individual choice and autonomy when it is that we're going to be working on cells. So for example, when they're doing the actual cell observations that we've been doing for the past week now, they do the observations and they have the options of alternating seating. So I actually seat all my students in groups as pods, as you can see, and I let them kind of choose their partner. And that really gives them some autonomy in how it is they work and with whom they work. I like to group them, kind of based on needs as I see it, but in particular, like with my students like Betsy, she has some students that she's really comfortable with, that she likes working with. So I like to give them that choice. And of course, like you saw in the warm-up options, like the different ways I let them show me their work. So that kind of gets a lot of buy-in from the student, because they get to express themselves in different ways that are really optimal for them. To also make sure that I am recruiting interest from the student for this guideline, I like to optimize the relevance. And in the interview I did with Betsy, she told me that she's really interested in nature and life. And so when we have these conversations like we do during our warm-up sessions, like I showed you in my notebook, what I like to do is I like to make sure that students know that this is something that applies to them, that it's relevant to them. And so for like Betsy, that was really helpful, because she loves learning about plants, she told me, and we're currently studying plant cells as well. And again, to help to ensure that I'm recruiting interest, I like to minimize threats, and the way I do that is, like I said, I do like to kind of mix it up. I do different task variations. So sometimes I'll have them write down what it is they're doing, or sometimes I'll have them draw or illustrate what it is that we're doing in regards to cells. So just kind of giving the option of mixing it up, of different ways of expressing what it is we're doing, I find really helps with recruiting that interest. What I also like to do to make sure that I am giving students options for recruiting interest is making sure that I have some sort of sensory support. I found that sometimes, students like Jefferson, who really need like that one-on-one support, he told me if he's feeling stressed out, I told him, hey, just take a breath or get up, walk around, and just doing that physical action, because he's a hands-on learner, it really helps him. Moving on to Guideline 8, Sustaining Effort and Persistence. What I like to do is make sure that I'm heightening the salience of goals, and the way I do that is with this progress monitoring. So like I want to show you again, I like to do these warm-ups. I use this as a way to make sure that they know every day when I do them, okay, you got to stamp that day. Okay, that means you understood that. And then, they get stamps every day to make sure they know where we're going. I post these, and those little daily stamps that they get kind of lets them know that they're on the right track. To also sustain effort and persistence, I like to vary the demands to optimize the challenge. And like I said, I sometimes have them illustrate drawings. Sometimes they're actually in their Cornell Notes, as you can see. I have them write down questions about what it is we're talking. They're always recording observations, or they're working on microscopes. And again, all this helps to make sure that the effort is sustained. I like to then make sure that I'm fostering collaboration. And like I was saying, when I'm doing my microscope work that they've been doing, they're looking at these unknown cell slides. And for some students, that's kind of difficult. They may not know where to find the information in their notebook, so then I have them work together in groups. They can do a think, pair, share, or a talk aloud with a buddy, and that really helps to kind of foster collaboration and just kind of build community. And for students like Betsy, doing these echo readings, or even just reading these cards aloud in her group, kind of helps to build this sense of community. The next checkpoint 8.4, which is increasing mastery-oriented feedback. I like to model, for example, how it is that we're using microscopes the right way, the wrong way to work in a science lab. I actually also give them an option to give each other group feedback. So I have a little contributor grading rubric that I embed within a lot of big assignments, which I can show you over here. So if I go into the student's website, I'll log you in over here to assignments. And I'm not going to show you the actual grades, but I'll show you how it is I let them know how it is they're doing. So go to assignments here. Sorry, I got a little bit of a lag. So let's see, where's my contributor grades? So there's the goals for success assignment, and there we go. Contributor grade for lab. So that's how I have them kind of get some mastery-oriented feedback, so they know what it is I'm working towards. Moving on to the last and final guideline, which is self-regulation. I like to give options for all students, not just my focus students, to promote expectations and beliefs that they can do this. And what I like to do is, not only do I give stamps to acknowledge that they're getting the answer correct, but I like to acknowledge actions that they do. Hey, Betsy Jefferson, good job. That's how you use a microscope. I like to, I use, rather than just calling out names, I do equity sticks. So I don't purposely call on Jefferson and Betsy. For example, I kind of just pull a stick randomly and call out a name, and that way that doesn't put too much pressure on them. What I also like to do is to facilitate personal coping skills. I have them work with a buddy. So like I said, Jefferson is a hands-on person, and he has requested of me that I give him some additional support. So I do partner him up with someone that he's comfortable with. I like to teach them a lot of like science, self-coaching mantras. So when we're gonna do a lab, I tell them, come on guys, say, I got this, I've done this, I could prove this with evidence. So that really helps with facilitating personal coping skills. And then the last thing that I do to make sure that I'm having students get some options for self-regulation is I do I have a way of developing self-assessment, which is my goals for success. I also like to do like one-on-one progress monitoring, especially with students like Jefferson, who was struggling earlier in the year, but now has picked up his grade. I like to just do progress monitoring, like shared progress monitoring between he and I found that's really helpful. And of course, I do like little check-ins, and I have them on exit tickets, just like reflect on, okay, what is it that I learned today? And so really for me, I believe that's kind of the way that I successfully was able to implement my entire lesson design. And again, we're still working on it, but I do believe it was successful. I now feel more confident in implementing UDL, and I believe that now for next semester, using this kind of approach by giving students these multiple ways of thinking, talking, and doing in science through multiple forms of expression, of action, of engagement and representation. I think that's really going to assist me and help me become a better educator. So I thank you for your time, and I hope you enjoyed the video. Bye.